In this video, we'll be talking about the Watson and Crick model of DNA. We'll be seeing about the basic features which were proposed by Watson and Crick in their model of DNA. So the first thing that we need to know while talking about the Watson and Crick model of DNA is the DNA is made up of two strands. And what do I mean by this? It means that the whole DNA molecule, which is a nucleic acid, is basically made up of two strings. This is one string and this is the other string. As you can find, I have just drawn a magnified structure of this here. Now, each of these strands is a chain of nucleotide. What is a nucleotide? Nucleotide is made up of a nucleoside plus a phosphate linkage. A nucleoside plus a phosphate linkage. Nucleoside, a phosphate linkage. All this together is a nucleotide. If I take one nucleotide, it means it is having a lot of nucleosides in it. A lot of nucleotides together will be found in one strand of DNA. So if I'm going to take this strand of DNA, it means it is having a lot of nucleotides in it. In a similar way, the other strand of DNA has also got a lot of nucleotides in it, chains of nucleotides. The two strands of DNA are anti-parallel. What is the meaning of this anti-parallel? Anti-parallel means when one runs from 3' to 5', the other strand runs from 5' to 3'. So to make you understand in a simple way, this is 5' and it runs along the line and it comes to 3'. While this end it is going to be 3' and it is 5'. So this is what is called the anti-parallel strand. The next one, the diameter of the helix. Diameter of the helix. Helix means if you look at the double-stranded DNA structure, the total length that it is occupying from either end of its bodily structure. This is called as the diameter. The total width that it is going to occupy is called as the diameter. This diameter of Watson and Crick model of DNA is 20 angstrom. And you all know angstrom is the unit that is used for measuring small atoms, molecules. It is a molecular SI unit. So you will be learning about more of that in chemistry. Now, each of the turn in a DNA helix is 34 angstrom. Now, before getting into what is the turn, we should first know what is the meaning of the word groove. Only then we'll be able to understand what a turn is. If you look at the model of the DNA that was proposed by Watson and Crick, you'll be finding at one portion there is a very shallow groove, while at one portion there is a very deep groove. What is the meaning of that? Shallow groove means if you look at from the central point of view, the distance between the central axis, if I'm going to draw as this is the central axis, the distance between the central axis and the groove, if it is very, very less, then I call it to be a shallow groove. If the distance between the central axis and the groove is going to be more, then I call it as the deep groove. Here you find there is no gap at all between the central axis and the groove. So I call it as a shallow groove. Whereas here you can find that the distance between the central axis, and the groove is long. So that is why it is called as a deep groove. So the distance between two shallow grooves or a two deep groove is called as a turn, which means the DNA helix will be turning or rotating like a circular staircase, and this will be exclusively happening only between two shallow grooves or two deep grooves. So you can take two shallow grooves or two deep grooves. The distance between them is going to be 34 angstrom. And if you take one turn, it is again going to be one helical turn. So one helix or one helical turn means it is between two shallow grooves or between two deep grooves. And between that one helical turn, they are going to be present 10 base pairs. What is the base pair? We have already learned about the nitrogenous bases. So the nitrogenous bases include adenine, guanine, thymine, and cytosine, and uracil as well. Since we are talking about DNA, it is only adenine, guanine, thymine, and cytosine. So if you take one turn of an helix, when you take one turn of an helix in a DNA, it will contain 10 base pairs. That is, it will contain adenine, guanine, thymine, and cytosine in a count of 10. Now, how will this be present? In one strand there will be a purine, in the other strand there will be a pyrimidine. And it can be the opposite also. In this strand it can be a pyrimidine and it can be a purine on this one also. But if you look at one turn, totally it will contain only 10 bases, or I can tell 10 pairs of bases. So how are these bases bonded, and what is the significant rule that these bases are being bonded? All these things will be seen in the molecular structure of DNA. Now the distance between the two consecutive base pairs. So I told you there are a total of 10 base pairs in one turn. The distance between two consecutive base pairs is 3.4 angstrom. So if the distance between two consecutive base pairs is 3.4 angstrom, then the distance between 10 base pairs is going to be 3.4 into 10, which is going to be 34 angstrom. Now, if I am going to tell between 10 base pairs it is going to be 34 angstrom, then that one turn, if you are going to take DNA, one turn of a DNA will be having a measurement or a distance of 34 angstrom. So to put all the three things together, the total length of the helix of one turn will be 34 angstrom. In one turn there will be a total of 10 base pairs, and the distance between a pair of base pairs, that is a two consecutive base pairs, the distance between a pair consecutive base pair is going to be 3.4 angstrom, and the distance of one helix or the length of one helix is going to be 34 angstrom. And one more thing which you should always know is the direction of helix is always clockwise in the model that is proposed by Watson and Crick. There are other models also which we'll be seeing in the further video. And if you look at the model that is being proposed by Watson and Crick, the pentose sugar, which can be ribose or the deoxyribose, along with the phosphate linkage, which together forms the nucleotide, will be like the skeleton. So they are the basic skeleton. So there will be a skeleton on this end and there will be a skeleton on this end. So the strands which we are talking are basically the skeleton. This is the skeleton and this strand is again skeleton. Each strand acts as a skeleton. Each of the skeleton is going to be bridged each other. They are going to be joined each other, and the chemicals that are joining them are purines and pyrimidines. This is very, very important. We should always have in our mind that the skeleton or the backbone is nothing but the pentose sugar or the phosphate, and purines and pyrimidines are the bases which actually join them as bridges. And Watson and Crick proposed this model of DNA by a technique that is called as X-ray diffraction technique. So I'll quickly sum up all these things. Watson and Crick model of DNA is the most traditional model even now followed for the DNA study. They told that the DNA is made up of two strands or two strings. Each of these strands contains a chain of nucleotides, and these two strands run anti-parallel. That is, if one is running from 3' to 5', other will run from 5' to 3', and the diameter of the helix, that is one turn, is going to be, the diameter of the helix is going to be 20 angstrom. That is the total bodily content or the width of the DNA. The next is each turn of the DNA is going to be having 34 angstrom. That is one helical turn, and it can be between two shallow groups or two deep groups. And in one such turn, you will be finding 10 base pairs. And the distance between two consecutive base pairs is going to be 3.4 angstrom. And the total distance of such a turn is going to be 34 angstrom. The direction of the DNA helix is always going to be a helical turn is clockwise direction. And finally, the pentose and the phosphate form the skeletal bridges, and the purines and pyrimidines together form the bridging accessories between the two skeletal bridges. So when you look at the last statement that between this formation of the phosphate linkage, the pentose sugar, and the purine and pyrimidine, how these four are interrelated, because we saw on a very simple overview that it is made up of two strands, and these two strands are going to be linked to each other. But how are these linked? What is the molecular structure behind these linkages? We'll be seeing about that in the next video.